Okay, hi, it's Mr. Kelly, and I want to offer. Some people said, "Hey, is there any other opportunities for grades?" And I feel a little guilty that you guys have been just doing exercise after exercise on Khan Academy, so I wanted to offer you an opportunity for an extra grade. It's not extra credit. What it is going to be a little lab that you can do, and if you have the materials at home, or you can substitute some of these materials and show me some results, then I'll give you a lab grade for it. Okay, what you need is a source of alcohol. I don't have rubbing alcohol at home, so I'm going to try to use this hand sanitizer, which is high content alcohol. The best bet would be to put your alcohol in the freezer for a little while and make it cold. This isn't cold, but we're going to see how it does. You need like a little bit of shampoo or hand soap, something like that. Nothing too crazy. That's nothing too crazy in color. I wouldn't do like bright red or something. You're going to need some sort of, normally would be doing a test tube, but this is the best I could find. If you have like a really thin glass of some sort, anything, candle holder, it has to be thin and clear. I know this isn't perfectly clear, but we're going to make do. I also use these. You don't have to have this, but because this won't stand on its own, I'm going to try to do this part. I figure this most sciencey part of this will be how well you can use the materials to be resourceful. Okay, so that has to hold that little test tube up. Okay, you need just a little bit of salt, regular salt, a little bit of water. I don't know. We're gonna say an ounce or two of water, and you need a funnel of some sort. You can make a funnel out of anything really. Like you could take the edge of this, right? And you could take another one of these. You could take like just some sort of, let's say like a Ziploc bag, for example, and try to cut the bottom of it and stick that in as a funnel. I'll show you what it's used for, so you decide. But you being resourceful during this lab is really the most creative part. Okay, what you're going to do is, you might have done this lab before. You're going to extract DNA from a food source. The lab typically calls for a strawberry, but you can choose a vegetable, a different type of fruit, something. And the whole point of like getting different results. The interesting part about what we'll post our results are what you can get DNA out of. But so you're going to take your source of DNA. All right, we're in the unit on DNA, and food has DNA in it, right? This is made of cells. This strawberry, so is a banana, so are peas, so are chicken. But I don't recommend handling raw chicken. But any living thing that you're eating contains cells, which means they contain DNA. So you're going to take a little bit of your food source, right? A piece of a banana, a bunch of peas mashed up, or whatever. You got mushrooms at home. Nobody's going to eat. Whatever. Try all sorts of different stuff. I'm not grading you on whether or not you get perfect results. I'm grading you on attempting it accurately. So I'm going to mash up the strawberry. I'm going to actually add a little bit of water. I would estimate this to be like, I don't know, maybe an ounce of water. If you think about a regular wax cup is probably 8 ounces, so 1/8 of that. Okay, just to make it a little thinner. I'm also going to add a little pinch of salt. Okay. Now, smashing up the strawberry itself breaks the cell wall. We're gonna add a little bit of soap, a tiny bit of soap. Okay, you don't need a lot, otherwise it's gonna get real foamy. Okay. Basically, the smallest drop you can get out of shampoo or something is enough. Maybe I'll add a tiny more water. Okay, zip it up. Okay, and then you want to get this to be kind of as evenly mushy as you can. You don't want to shake it up and make it foamy because then it won't go through. But it's okay. Now it's so the smashing it with my hand breaks the cell wall. Okay, if you want to blend it or something, you can, but it's probably overkill. The soap dissolves the cell membrane. That's why we use soap for stuff and detergent for stuff, because it dissolves stuff like oil off your hair. It'll dissolve, dirt off of pans. It's soap, just like phospholipids, is called an amphipathic molecule. It's got a polar end to dissolve polar stuff. It's got a non-polar end to dissolve non-polar stuff. Okay? Now, we're going to take this whole setup and we're going to put it through our little quote-unquote test tube apparatus, which actually I really needed to seal off at the bottom. Give me 1 second.